Welcome to Itasca Water's Practical Water Wisdom Series. I'm John Downing, your host for today. Itasca Waters is a nonprofit whose goal is to educate residents in Itasca County and the region about how to keep our water clean for both the environment and the economy. We have been teaming up with like-minded people and organizations for 14 years now, and you can read about those projects on our website at itascawaters.org. If any of that is of interest to you, please consider becoming a member or a volunteer to help us support programs like this. And we thank our partners shown on your screen for their support in producing this program. On this next screen is your guide for today's program. Dr. Cody Sheik will speak for about 30 minutes or so, and then we'll answer questions from you. To submit a question, you only need to click on the Q&A button at the bottom of the screen and type in a brief question. You can do this at any time, and we'll read the questions on your behalf. We will not be using the chat function today. This program is being recorded and will be available for viewing online through our website, itascawaters.org. Finally, we value your opinion, so please click on the survey link, which will be on the screen at the end of today's program, or complete the survey that will be sent to you by email. We thank you for doing that. Today's presenter is Dr. Cody Sheik, Associate Professor at the Large Lakes Observatory, University of Minnesota Duluth. Cody is a microbial ecologist who focuses on the role of organisms that are just too small to be seen with the naked eye in lakes and other waters. He's especially interested in blue-green algae, which are called cyanobacteria, and the role they play in freshwaters. Cody's lab uses DNA-based approaches to identify microorganisms and the functional roles they mediate in the environment. His work is primarily focused on the Great Lakes, but recently has extended to blue-green algae blooms across Minnesota. Cody uses the DNA of the cyanobacteria or blue-green algae to identify those that produce toxins versus those that are not poisonous, with an ultimate goal to identify the tiny but dangerous culprits. Cody's topic today is impending aquatic invasive species threats. Can they kill my pets or hurt my kids? Microorganisms, that is, those that are too small to be seen without a microscope, are an important linchpin of all ecosystems, but the overgrowth of bacteria can lead to short-term and long-term problems for lakes. Certain bacteria, specifically the cyanobacteria, can produce toxins which can impact human and animal health. In this talk, Cody will share important research they are doing across Minnesota and the Great Lakes to identify what microbes are present and to consider the implications to our environment and our use of water for recreation and daily living. He will also talk about some non-native microbes that can cause even bigger problems as they invade our lakes. So it is a real pleasure to turn the microphone over to Cody. Cody?
Thank you all for having me. I really do appreciate this invitation and to get to share some of the work that we've been doing in the lab with you all and think about a topic that has really been growing in my lab here of recent. And this is thinking about cyanobacteria as invasive species. And I think, at least for me, when I started thinking about invasive species, the biggest ones I started thinking about were some of these common ones that we see, things like zebra mussels, the spiny water flea, or even Eurasian water milfoil. And, these are sort of these poster child ones because, they're quite charismatic in terms of megafauna, right? We can see them. And, in terms of zebra mussels, you can see these beds on the bottom. If you're fishing, you might see spiny water flea attaching to your fishing line, and Eurasian water milfoil can really change the landscape of a lake or a watershed, and that's really why we're really important or really focused on some of these important aquatic invasive species. Is that they do have the ability to change the environment and change nutrient flows. So thinking about food webs that will ultimately impact water quality and also fishing and recreation in not just Minnesota waters, but waters across the world. And so these poster childs really stand out for a lot of us. And in Minnesota, the Department of Natural Resources and also through the university, the Minnesota Aquatic Invasive Species Research Center, are really focused on one, identifying pending threats, so stuff that may be coming in, but two, also trying to think about the distribution of organisms that are already established here. And you can see here these QR codes, if anybody wants to take a snap with their phone and learn more about these AIS from the DNR or from MACIRC, please feel free to grab those real quick on your phone. And I also have the links down here if you're interested in those as well. But what we're looking at is 9 to 10 different fish species, 10 to 12 different plant species, these invertebrates like the spiny water flea, we have around 10. But we also have this microbial or viral fraction here. And being a microbiologist, this is one I was really interested in and looking at as we started to develop our research program. And when we look at these impending threats here, a lot of these have to deal with fish and or fish habitat associated problems. And so a lot of these are like tapeworms or these lymphocytes or sarcomas that these organisms are causing are really impacting our fish populations, and from not just the sheer numbers, but also harvest and quality of the fish that we're catching. These can all have a really traumatic impact on these. There's also, we're also doing work here on this organism called Didiosemia geminate, which is also recalled as rock snot. And I'd love to talk more about that today too, but I think for sake of time, I'm gonna put that off to the side. But that's also one of these microbial impending threats. But when I look at this and think about these, some of these are viruses, some of these are microorganisms, but it's really a small fraction of the total diversity of microorganisms that we should maybe be considering. And so this is really just a small fraction of life on Earth as we know it. And this is just the current tree of life that we have right now. And we do this using DNA-based tools. And what you can see up here is just there's a huge diversity of bacteria up in this very top part. And this always just like really blows me away every time I think about this, is that you see these little individual black lines coming down here. That is at the order level. So if we think about kingdom, phylum, class, order, family, genus, species, this Linnaean way of naming organisms, we're really down at this order level, right? Just right below phylum. And what gets me is that if we blow up these lines here and actually look in at each one of these little leaflets here, we would see hundreds, maybe even thousands of species within each one of these lineages and these lines here. And that really gets me because I really like to think about just diversity of microorganisms and what's causing things. But when we compare this over to, say, this eukaryotic branch where we are, it's quite small in terms of the overall diversity. And that's not meant to diminish eukaryotes in any sort of way. It's just one of these things where this unseen majority, right? And a lot of us eukaryotes, not just us, we have a microbiome on our skin and in our guts that we have to maintain. And every animal does as well. It's just that we rely on this unseen majority of organisms every day. And mainly because we can't see them, we tend to forget about them. And so this is up here just to show you some of the diversity that we see in the microbiome itself. So this is probably a piece of fiber from a salad or something this individual ate. And here you can see up in this upper right-hand corner, all these different organisms that are shaded in different colors and shapes and sizes. Some are very small, some are quite big. This is the yeast over here. And then when we're in the lab, we can try to cultivate these organisms or grow them. This is just an agar plate that has just some glucose in it, I think, and some other nutrients. And you can see this comes from soil, and we can just see the total amount of diversity. There's different colors, there's different shapes, and these plates are about this big around. And so you can kind of get a scale for size here as well. But we rely on these microorganisms all the time, and most of these organisms are beneficial. And so here in this panel, this infographic here, really the ones we're worried about that cause diseases are here in red, right? So there's only 4 of these. And so you can see that they're a very small majority of the total organisms that we need every day. So we have organisms, aquatic organisms that we rely on, water bodies like cyanobacteria hanging out in the water are producing oxygen, they're making carbon, things like this. We have organisms that we rely on for food, right? I don't know if anybody ate a yogurt this morning, but you ate millions and millions and millions of little tiny microbial cells that made that yogurt. And so we rely on these organisms all the time, and we tend to not really even think about it. Most of our plants have these really delicate symbiotic relationships with the microorganisms. If you're growing beans out in your garden right now, there's nodules on those roots. And inside those nodules, there's microorganisms that are taking nitrogen gas from the environment and making ammonia and feeding the plant. And so when I think about the total flora of some of our aquatic scapes, rivers and estuaries and lakes, it's really thinking about what is sort of a natural state of some of these lakes. And that can be quite hard because, as you'll see later on, we've been changing these environments and shaping these environments for hundreds and thousands of years in some cases. And so part of thinking about, what does a diseased lake look like versus a healthy lake are some of these impending questions that we have that really directly relates to these aquatic invasive species. And so my lab has really been focused on different scales and thinking about microorganisms. I think our motto right now is small microbes, big environments. This is Lake Superior in the background. And when we go out on Lake Superior, this is aboard the Blue Heron. We're going out and we're sending this instrument down. And this instrument's called a CTD. It measures conductivity of the water, so how much stuff is sort of in the water. Temperature, and then also depth. And these bottles here allow us to actually collect very discrete packets of water that we can then bring back into the lab and analyze. And so we use this instrument a lot in different forms. And when we're at small lakes, we use these just very small bottles and that allow us still to collect water. And when we go out in there, we may see cyanobacteria like this one up here. This is Dolichospermum linomanii. And it forms these really dense balls of cells. And you can see actually individual cells there. These are actually really, really big cells for this organism. But you can actually see these colonies suspended in the water just by holding it up if you had like a glass or like a water bottle. And you can actually see them floating and moving up and down in the water. But this graphic down here you may have been focused on is really showing you sort of this different sort of scales that we're working at. We're going from the big environment and we're thinking about these colonies, but we're also thinking about these very small single cells. And so what we have here in this SEM in the very bottom right is a zooplankton, which is just natural out there in the water. They're chomping away on food and they're starting to process a lot of this organic matter. We're going down to a diatom here, this big circle. It's a phytoplankton. They're doing photosynthesis and they're in every natural water. But then we have these little single cell bacteria. That's this little rod there. That it zooms in on. And these organisms can be both attached to other organisms like it is here, or it can be free floating out in the environment. And what always gets me is just what are these organisms doing? And we really try to use different tools and techniques to really get at this. And I'm a microbiologist, so I'm already biased here, but I like to think these organisms as quite mighty. And that's because they really mediate these very important geochemical processes. So one that's really important right now to Minnesota is sulfur and sulfur deposits of ore. This is fool's gold here. And we have organisms that can take that fool's gold, which is also called pyrite, and it loves eating this iron and this sulfur off of it. And when it attacks that sulfur, it turns it over to sulfuric acid. And that sulfuric acid can then go on to do a thing called acid mine drainage. And these organisms just love doing this one process, and they're really, really good at it. But they can have very devastating impacts to the environment. One that we've been looking at here lately is at Lake of the Woods. This is a seasonal cyanobacterial bloom. I think this was taken maybe last year from Cyanotracker, and you can just see the extent of this bloom across the lake. This is done by an organism called the phanosomenon. But these organisms, these cyanobacteria, they photosynthesize, and so they have chlorophyll, which makes them green. But they use the energy from sunlight to really take carbon dioxide or CO2, fix it over into sugars that it can use for biomass, takes in some nitrogen and some phosphorus and grows, and produces oxygen for us to breathe. But it turns out these cyanobacteria are also really, really good at synthesizing different chemicals that can be quite potent for people like us. And so these mighty microbes, as I was saying, are very good chemists, and it all deals with their genes. So each one of these organisms has a genome, a chromosome, and on this genome, there's genes within there. And these genes encode proteins or enzymes that can then go on to catalyze very specific reactions. And when these organisms put these different reactions in sequence, so going from like reaction 1 to 2 to 3, they can synthesize these really, really important compounds. And so my lab has really been thinking about genome sequencing, where we can go in and sequence the genomes of these organisms using some DNA sequencing tools, and really start to investigate these organisms from a very primary state, thinking about what does the DNA tell us that these organisms are capable of. And so for cyanobacteria, we've been really thinking about these toxins that they're producing. And cyanobacteria are able to synthesize many types of these secondary metabolites. But we're in Minnesota, and we're very interested in right now, from a toxicity standpoint, are these 4 primary ones. We have this one called cylindrospermopsin. We have these things called saxitoxins. This is also known as paralytic shellfish toxins. So if you eat some bad shellfish, it can go bad for you very, very quickly. Anatoxin is a very important one. And the most prominent one that gets a lot of exposure are things like microcysteines. And these things have various states, and they attack the liver. And so we're really focused in on these because they can cause quite a bit of problems downstream for humans and also our pets. And so those anatoxin and the saxitoxin are very potent toxins, mainly because they're neurotoxins. And so under sort of light exposure, you may get some headaches, some drowsiness, maybe a bit of numbness in your mouth. But if you have a high enough dosage of this, eventually it causes muscular paralysis, and which can then shut down your lungs and also your heart, right? And so things can go south very quickly with some of these toxins. Things like microcysteines or these cylindrospermopsins, if you're ingesting these, they can really cause a lot of gastric problems, like nausea, vomiting, diarrhea. And the higher the dosage that you see can really start to go after your kidneys and also your liver, okay? And so when we're out interacting with our waters, we have to really be cognizant of some of these compounds that may be present. But also, we need to be cognizant about our pets. And pets are usually are the more leading cause of deaths for some of these organisms, for some of these toxins. I think we see some of these several times a year, especially here in the Midwest. But your pet is also susceptible to these, and so it can cause vomiting, fatigue, shortness of breath, difficulty breathing, same sort of stuff that we would see here. And because sometimes they're much smaller than us, they can receive lower dosages and actually cause death much more quickly than us. And so we really need to be really wary of these as we move along. And so myself, Dr. Chris Filstra, Dr. Schreiner here at the University of Minnesota Duluth, have started doing a small project that we're starting to wrap up, I guess, right now, funded through Minnesota Sea Grant, where we went out and started looking at lakes spread across Minnesota. And we have a couple lakes down here in the south, Cary and Peltier, and Portage and Hill in the middle, and White Iron and Tate up here in the northeast. And we've been trying to do a very holistic sampling approach where we go out and look at nutrients, we look at toxin concentrations, some microbial processes like nitrogen fixation, do some taxonomy with a microscope, but also with these DNA tools. And then we also go out and look at these microbial functions, doing some other sorts of types of DNA or RNA sequencing. And so we've been really looking at these lakes and trying to think about, what's the distribution of cyanobacteria across these landscapes? What are some of the controls on their growth? And importantly, are they producing toxins? And so one of my students, Kayla Natwara, has really been looking at the DNA portions of this. And she's been going out with these DNA sequencing tools and looking at the distribution of these cyanobacteria across these different lakes and across different years. And over here, these bars just mean the bigger the bar, the more abundant these organisms are in our data sets. And the colors indicate different species. And I'm not going to give you a test on these different species here, but I'll highlight a couple as we go along. And so some of these green bars that you see that are very abundant in some of these lakes, most of them, are these cyanobiums. And these organisms are really important for not just producing oxygen for us, but also fixing CO2, making biomass, and really forming sort of the base of some of these food webs. Some of the ones that we are worried about are this, like say, this burnt orange one here, this microcystis, or even this dolichospermum one here, which do have, in some cases, have the ability to produce toxins. So when we start looking at the landscape of what's there, we can start looking at these very specific lineages or clades of organisms and start to narrow down maybe who is actually present and how abundant are they, and whether or not they're making toxins. And so my student Kayla, again, has been thinking about these different linkages. So we see the broad base of who's there, but are we seeing the same species or same strains spread across these lakes? Or because we have such a huge gradient in distances, are these lakes sort of isolated and they'll have their own populations? And it turns out that it's a bit of both. And so all 6 of these lakes actually share 47 species that are spread across these things. And when we look at the distribution of who they are here, we can see again, the cyanobium is this nice orangey color here. We see a little bit of that dolichospermum in this light green as well. But we also see this microcystis being this pink one that we're really keyed in on and starting to think about, primarily because it has the potential of making this toxin microsystem. But you can see also some of these lakes have very unique cyanobacteria that are associated with them, with just the lake itself. And so we're really trying to get this big landscape and trying to discern what these organisms are doing. And so right now we're in the work of actually identifying who's producing these toxins and really trying to dive into some of the data a bit more. And so check back later on, we'll have hopefully some papers coming out here in the coming year or so. But again, we've also been measuring concentrations of toxins, and we've actually measured microcystin, anatoxin, and cylindrospermopsin across all of our lakes. And so microcystin here is a solid line. We have anatoxin is this dotted line here, and this dashed line just means cylindrospermopsin. And you can see we have lots of different ranges of concentration. So in Peltier Lake, which is one of our southern lakes, which is right on the north side of the Twin Cities, it's very eutrophic, has a lot of nutrients, and we can see there's a lot of microcystin being produced in this lake, but we also see a lot of anatoxin at very specific times being produced. And so the big takeaway here is that in most of these lakes, we are well below the concentration, which I've labeled out here, that Minnesota has set for water quality standards for swimming. So microcystin is at 6 micrograms per liter. And you can see in most of these lakes, we are well below that, except for Peltier. So if you're planning on going to Lake Peltier anytime in the next month or two, just be wary and maybe not swim and maybe not keep the fish out of there as well. I'd be a little bit wary of that. But you can see for the most part, we're at a very low concentration of these toxins, which is really great, right? And so we've been trying to dive into a little bit more of this data and trying to think about, could nutrients be driving toxin production? And that's because there's been some really great work in the past showing that compounds like phosphorus, or the combination of phosphorus and nitrogen, both impact the production of cyanobacterial biomass, and then also can influence toxin production as well. And so these are some of these really foundational works here. This is up in Canada, where they split this lake into two using a barrier, and showed they did some fertilizations here and here, and really show that this phosphorus treatment really ups the amount of chlorophyll that they see. And so when we look across our data set, we have total phosphorus here is on the x-axis, and total nitrogen is up here on the y-axis. And the size of our dots here just represent how much microcystin is present in that sample. You can see that when we're under these nitrogen limiting steps, so we have both nitrogen and phosphorus, but the ratio of these two is in this sort of stress state where they're nitrogen limited. And it's under these sorts of conditions is where we actually see microcystin being produced the most, rather than in some of these other more balanced lakes that we actually have. And so it seems like there's a really complex role here. It's not just a one-to-one play where we have phosphorus and nitrogen. We should have more of these toxins. It seems like there are some other factors at play. And so one of the big things that we're looking at is that nutrients alone may not be driving this growth. And when we look at the total impact of the lake itself, so each one of these points here represents one of our samples, and the closer these points are in physical space, the more similar they are to each other. We can look at some of these driving factors for some of these different lakes and different samples. And we can see for lakes like, say, Cary, it's about dissolved organic content month, the watershed lake, and so, or the water stability of the lake here. While some of these other ones are more about average temperatures, ammonia, percent of agriculture, nitrate concentrations. And so it looks like these lakes are potentially diverged and are responding differentially to these different stimuli, like nutrients and things like that. And so one of the things that we're having to think about now is, like, will these cyanobacterial populations all respond very similarly, or will they have their own trajectory over time as things change? And so that's just mainly for Minnesota, but we have all this great body of literature out there that says, you know, all these nutrients and things are sort of like a part of the story, there are other things at play here. And one of these is temperature. And so this is just showing lakes that are responding positively to increases in temperature, and then also phytoplankton blooms. And you can see there's a lot of red or light pink shading across the earth here. And that's really the big take home point here, is that there's a lot of change that is happening in these lakes. And I don't know if anybody has paid attention or not to Minnesota lakes, but the Minnesota Department of Natural Resources came out with this finding where they've been tracking temperatures of the lakes and ice in, ice out, and have found a very significant change with time. So dating back to when these things started being taken back in 1896, we can see that we've been on this gentle and gradual increase in temperature. And because of this, lake ice season has decreased on an average of 14 days. And here you can just see it's different for different types of lakes. So Lake Bemidji is like almost 19 days, while lakes like Detroit Lake are only about 9.5, maybe 10 days. And this is a product showing down here at the very bottom, and just store these changes that are across the state. Some of them are more impactful than others. So in this very central northern portion of Minnesota, we've seen about a 4-degree increase in temperature, while over here by Duluth, we've only seen about a 3-degree. And then we're also having big changes too in annual precipitation. You know, a lot of the state has been in drought this year, as many people have seen, but it can be very patchy, right? And so precipitation patterns, which lead to runoff events and things like that into the lake, the watershed, as it changes because these precipitation patterns, because of this temperature, they're all going to have really big impacts on the lakes and the organisms that reside in those lakes. And when we think about temperature just in general, we see that globally lake temperatures are increasing, and Lake Superior is on this as well. But there's a big coupling between microbial growth and temperature. And these organisms have evolved over time to be very specific about the temperature ranges that they like. And so over here on the right-hand side, we can see two different species, an Aphanizomenon, which is like the ones that are in Lake of the Woods, and this Microcystis aeruginosa that we find kind of everywhere in these Minnesota lakes. Both have these temperature optima that they like to grow at that are quite different. This one is about 15 degrees, while the Microcystis is more of about 30 degrees Celsius. And so with our changing thermal landscape of these lakes, the timing at which these species are, these cyanobacterial species are maybe increasing their growth or potentially overtaking another organism over time is going to be changing. And so we're going to be seeing the landscape from what I just showed you, the big picture of all those cyanobacteria. We're going to be seeing this landscape change over time, and which is frustrating and also exciting from a research standpoint, I guess. And so should we be worried about that? And I think, yes, we should. There seems to be some evidence that chronic moderate dose exposure, so like through drinking water and things like that, can lead to liver and kidney damage. And so this is just some new work that's been coming out this in 2017 showing that it really is there's a breaking point about the concentrations in mice where you start to see these significant changes. But also, this is mainly just for liver, but for things like the kidneys, it's a little bit more sensitive. And so the concentration and dosing for the kidneys seems to be much lower than it does for the actual liver. And so there may be these differential effects that are happening for different organs in our body as we're being exposed to these. And so some of these big questions here as we start to wrap up is thinking more broadly about how do I know if it's a bloom and kind of what should I be doing about it? And, you know, for a lot of these invasive, not just cyanobacteria, if you see them, you need to say something, right? So if you see spiny water flea in your lake and you've never seen it before, maybe call your local DNR representative and tell them, maybe take a sample if you can. And just getting the word out will actually filter down to us as researchers eventually. And I'll talk a little bit more about this. And so, you know, if one of these things is that this was just at a local lake here, they came out to go fishing one day and then they saw this. This is a cyanobacterial bloom. It's kind of this really thick green, but you can also see they're called blue-green algae, is that in some cases, some species produce these really bluish, beautiful green hues here, as you can see in this water. And so if you see something like this and it's out of the ordinary, definitely give somebody a call about this and report it. These blooms can come in all sorts of shapes and sizes. So this one is one that happened here in the Duluth Harbor. It was very rapid. Basically, you blinked and it's gone. It just moved along with the water. Same thing with this one. This was a bloom in Lake Superior, out on one of the sandy beaches out there. And in these blooms, we can see that sometimes there's quite a bit of different, quite a bit of diversity within this. And they form these really beautiful shapes and sizes within here. But right now it's really tough because we have all of these different organisms in here. It's really pinpointing down which ones are potentially making toxins. And this bloom here actually was making microcystin at the time. And so these things come in, again, all different scales and shapes. This is a very small little bloom. It kind of looks like pollen, but these things are just little tiny colonies that I was talking about earlier on. And then you have these other bigger ones. I think this is Peltier Lake here. You can see how thick and bloomy and green it actually is. And this is over here is Lake of the Woods. This is way offshore at one of our monitoring stations. And so, you know, for me, when I go out and look at water, it's really about being observant. And for those of you that maybe live on the water, you probably make these things, right? Subconsciously, you're like, oh, the water looks a little different today, maybe. Or you notice little changes like this. And so noticing those changes is quite important in letting us, like myself, or John, or Dr. Downing, know about these things, like gives us awareness to them and know that we can, oh, maybe we go look at this sort of stuff. Blooms can also be easily mistaken for early season pollen events. And so Early on in the season, again, trees come out, we all get sniffles, at least I do, and we can see these different styles of tree blooms happening. And we get calls about this all the time here in Lake Superior and in the harbor and other small lakes. And these are just fine. Like, there's nothing wrong with this. You can go out and swim. You might be a little yellow from this, and you might get a little sneezy, but that's not a big deal. And so understanding sort of like these differences between what an actual bloom looks like and maybe some of these pollen events and things like that will help you realize as you move on what is safe and what is not safe. But again, a lot of this AIS detection, we can't be out on every lake, all right? Our license plates say we have 10,000 lakes. Actually, I think it's more than that, but we can't always be out there every day on these lakes. And so I would really like to highlight that we need you to be our eyes and ears on some of these lakes. And we can receive some of this data through different ways, and iPhones and just phone technology in general has gotten so good these days that there's actually apps out there like iNaturalist. There's a Bloom Watch app. There's one called Bloom and Algae. And these are all phone-based apps that will give you location that you saw a sample, allows you to take pictures of that sample and upload them to the internet, and they can be used as these database repositories. And we can generate these beautiful maps and things. I think this is for milfoil distribution through iNaturalist, right? And so you can see where these things are located in physical space, and really helps us as we start the next project thinking about, oh, what lakes, what areas should we be really concerned about for these different species. And I'd also like to guide you all over here to this USGS field guide and laboratory guide for thinking about freshwater cyanobacteria and flower blooms. And so if you're interested in learning more about what does a certain bloom look like, what does a certain species look like, really go take a peek at this PDF. I think it's about 50 pages long, and you can take a picture here and use that again with your phone if you wanted to. And it should take you straight over to that PDF so you can take a look. But definitely get informed. If you're really interested in this and understanding more about it, definitely think more broadly about it and trying to report to us, I guess I should say. And so kind of to summarize here that these cyanobacteria that we are looking at, most of them are integral for our food liquids, and we shouldn't be worried. They're doing their thing. They're helping the environment, making oxygen. They're doing what they need to do. And it's just really these select few that can actually produce these cyanotoxins that we really need to be worried about, and are the organisms that my lab is really worried about, thinking about their distribution and are they moving in different areas differentially across Minnesota. Unfortunately, with climate change happening, we're going to have to get used to these blooms and thinking more broadly about it. And so, informing ourselves is sort of like the first sort of area of protection. So, when should we be swimming, right? And when should our pets be out there? If it looks suspicious, maybe use some caution. It's sort of always my first take on this. But we're going to have to learn how to work around these blooms. And there are some things that we can start to do actively. One of them is just like we would for any sort of other AIS, like aquatic invasive species in Minnesota. If you're boating in between different lakes, please wash your boats, clean them out really well, so that we're not spreading these organisms from lake to lake to lake. And this is, we have these voter cards through Minnesota. This also applies for microorganisms as well. And if you're living on a lake or visiting lakes, try to always minimize your impact. Clean camping, not dumping on waste and things like that near the shoreline. If you live on a lake, maybe limit the amount of fertilizer you use. Thinking more about a natural landscape. It's also good for insects and things like bees. Limiting the amount of herbicides that we use. A lot of these herbicides actually contain phosphorus within them. And so as these things get broken down naturally in the environment by other bacteria, they can then be released out and help stimulate these blooms. Also, if you have a local sewage system, maybe check and see what the concentrations of some of these fertilizers or potential nutrients like nitrogen and phosphorus are coming out of here, and seeing maybe is there a way we can mitigate this from directly moving into the lake system as well. And so that some of these point sources are really big drivers of nutrients that these organisms need to grow and need to bloom. And so, with that, I would say, if you do see some blooms, definitely email me or give my phone a call. It may go straight to voicemail, but I'll get back with you. And if we know about these things happening, then we can actually start thinking and acting on them and going out and studying them. So with that, I'll leave it there. And thank you again for chatting, being here and chatting with me. So I appreciate it. So thank you very much.
Hey, thank you, Cody. Now let's go to the question and answer portion of today's program. It's going to be hosted by Bill Granches, the director of the Itasca County Aquatic Invasive Species Program and a longtime board member with Itasca Waters. Bill?
Well, thank you. Hello and welcome to today's Q&A session for the program. Now, remember, please submit your questions in writing by clicking on the Q&A icon. We're not using the chat function here. So now I'm going to read the questions aloud and they will be answered by Dr. Sheik. We're probably going to combine some similar questions and we might not get to all of your questions. So please feel free to send us an email after the program and we'll be happy to answer any of those questions. Okay, so let's begin. Wow, a lot of questions. Okay, are there good bacteria or viruses that might invade our waters and do good things, not bad?
Definitely. I would say that 99% of the bacteria and viruses that we encounter every day do nothing for us, right? They kind of pass by and they don't cause any disease to us. And we need a lot of these bacteria in the water system. So they break down carbon, they feed nitrogen to the system that we naturally need. And so, yeah, I think there are a lot of organisms that that we see coming and going that are great for the environment. And so one of the problems that we have is identifying these. We don't really have good records of nearly any water system. The best water system we have is Lake Mendota in Wisconsin that has this 20-year time transect that they've been monitoring this lake. And for most systems, we just don't have that. So knowing what is a good organism versus a bad organism for a specific lake is actually really tough. But I would say the majority of these things are good for us, so and good for the lake system.
Good to know. Can blue-greens turn off and on their toxin creation?
That is the $1 million question. They can, and it seems like for some strains it's different. So some of it is in response to nutrients. Some of the times it's in response to temperature or time. But there's really no great consensus yet on when these organisms decide to start producing toxins. And these organisms have been on Earth for about 1 billion to 2 billion years. And these toxins have been around because we can look at the genes, we can age these genes. They've been around for a very long time. So we know that they had functions, and these functions have likely changed as, say, eukaryotes came around, or as conditions, as oxygen concentrations have changed on Earth. And so their roles may be different than what they were 1 million years ago, if we think about it in that sort of way. But that's one thing that we're trying to do with these studies is figure out what these triggers are. Because if we can figure out that trigger for that very specific lake, then we can start issuing warnings, right? And just saying, hey, maybe don't swim here because we predict that there should be toxin production happening at this lake. And so that's one area where we see this research that we're doing growing. It's trying to think about these individual triggers for these different lakes.
Very good. Next question. Are there exotic blue-green algae living in excuse me, are there exotic blue-green algae that are invading our region here?
One of the big ones that we're really worried about is Cylindrospermopsis. And so this is a species that was typically more in southern extent, middle part of the United States. So thinking about like Missouri, Iowa, kind of below that. And we've been seeing an increase in this organism in Minnesota waters, and it seems like it's moving around quite a bit. And so this is one that we are worried about, mainly because it will produce that toxin, which it's named for, the Cylindrospermopsis, or Cylindrospermopsin is the toxin. Cylindrospermopsis is the organism. It's a mouthful, but that is one that we're really interested in. And Minnesota's MACERC, through the university, that's one of the listing ones that they have as potential future AIS species in Minnesota.
Okay. Wow, here's a good one. How many pets are killed during the summer in Minnesota, and is it a big problem, and how can we avoid it?
I don't know that figure offhand of how many it would be. A lot of times they make headlines though. It'll be like, you know, pet killed, and it'll make the local newspaper. And I should have tried to look that up. I apologize. I would say it's not zero and it's not thousands, sort of thing. But it is happening, and it happens more than what we probably would like, right? And so I think being cognizant of these things. So you saw sort of like the blooms, like say, like, I don't want to keep calling out like Peltier, but that one it's really green. It's a very highly populated lake. There's a lot of people that live on that lake and use that lake. And, you know, knowing that it can bloom, basically all summer and produce toxins most of the summer, you know, that would be one of these lakes where I'm like, maybe we don't go out and throw the ball for the dog and have it running on the water. But for the most part, it looks like a lot of these lakes, at least the ones that we've been studying, you know, there's going to be very little exposure for these animals in terms of the ecosystem. And so I think for right now, we should take that as a win. But, you know, again, if the waters look a little funky and are nice and green, and when they haven't been before, maybe we just don't throw the ball next to the shore and keep the dogs back from it. So that'd be my biggest one, is just, you know, use common sense.
Well, it's a tragedy for a pet owner when any pet is lost that way. Speaking of dogs, is it okay for dogs to drink out of lakes?
Yes and no, right? So we know that there are other sorts of, in terms of cyanobacteria, we don't want them drinking green, chock-full microsystem waters. But there are other sorts of organisms that live in waters that do cause gastrointestinal problems. Some of these things are called coliforms. So thinking about E. coli, right? So it can be, it's one of these things, it's a fecal contaminant. And there are several of these fecal contaminants, and they can interact with pets and us in different ways. And so, you know, it's, I don't want to say they shouldn't, but I try to limit the amount of pond water that my dog drinks, just because I don't want to have to clean up the mess later on.
Oh boy.
You know, having dehydrated too. That's another big part that can happen with the gastrointestinal things. So.
Very good. Thank you. Next question. If it's a windy day with lots of waves and a bloom is not obvious, can a bloom still be present?
Yes, and what a lot of times what will happen is that wind will push it against the shore, and that's usually a lot of times is when we'll actually see them, is that we'll have a high wind event and it pushes those organisms that could have been offshore or maybe just underneath the water, and it's pushed them over towards the shoreline, and that's when we see it. And so that picture I showed of the dock when they came out the next morning to go fish, that's probably what happened, was that bloom pushed around. We see this a lot in Milwaukee Bay. Some of my colleagues are out there, they have buoys. There's a big swim beach there, and they can actually watch these things move back and forth over time during the day. And so they actually have a buoy that has a camera on it. And when they start to see chlorophyll concentrations come up on their sensors, and they take a picture of it, and they can sort of issue warnings for people at the swim beach on this sort of thing. And it all has to do with the way the wind's blowing and things like that.
Okay, thank you. How do you figure out which microbes are present, and how do you count them?
So one way to count them is just through a microscope. So you take a little sample and we put them on a little gridded microscope slide, and you just sit there and tediously count them. It's like 1, 2, 3, 4, 5. And you can route that back over to how much volume you took from the sample. There's other more sophisticated ways of doing it. Some of it's called flow cytometry, where you take a laser and you pass water with the organisms by it, and the laser actually count these things. Some of them are, there's another one called a flow cam where it actually takes pictures. And so it dilutes it down and takes a picture of these organisms as they float by, and they get a total number that way. And we can also go about this doing it DNA wise. And so we know that there are marker genes that we can amplify, and we can count the number of amplified products off of that using just some molecular tools in the lab. And so there's multiple ways of getting at it. All of them take time. The counts are probably the quickest thing that we can do. And you can actually have this deployed out in the field potentially and say, oh, look under the microscope and it's like, oh yeah, we see a microcystis colony here. We see 15 of them in this water. And so that's usually the microscopy is the best way of doing it for initial things.
Okay, thank you. Next question. Can you always see cyanobacteria when they could be dangerous?
No, they can be very dilute in the water column. And so like you may only see like one colony per milliliter, but that one colony could still be producing a lot of toxin. And so we call this like a toxin cell quota, where sometimes you have a high density of cells and a high density of toxin being in this one colony of cells. But the overall number of cells within that water can be quite dilute overall. And so there's this imbalance that can happen. We can have a lot of cells, but no toxin being produced at the same time. And it goes back to the whole problem of like, we still don't know what triggers toxin production in many of these species.
Next question. Actually, this comes up quite a bit here as the water is warming and the temperatures are warming. We get so many calls about people asking us to identify different things that, of course, usually everybody thinks anything's a zebra mussel, but also they think it's cyanobacteria. But why do microbes do better than other organisms when the water gets warmer? And wouldn't warm weather help aquatic plants and other algae grow too?
That is a very good and complex question. So many species, especially here in Minnesota, our waters have been traditionally cold, right? And so we have these very cold adapted species that have this very small limit of thermal growth where it can do it at its optima. And it turns out for most organisms, once they reach that temperature optima, it drops off very quickly on the actual temperatures they can withstand. And so like bacteria like E. coli, and some of these cyanobacteria that you saw in that curve, they peak out and then they drop straight off. And so when we get to these, when we start pushing their thermal maximum that they can actually go and survive in, a lot of times it turns them into like stress and survival mode, right? Just like right now, when I go out to walk outside, it's 90 degrees outside in Duluth right now. And it turns out that's like my thermal max, where I'm just like, I can't really think or do anything. Same thing happens with these organisms. And for some of these plant species and cyanos and other organisms, they just get stressed out. And eventually what we'll probably see is a replacement of organisms as these things move against these different waters, right? So we'll get to see these organisms that have a higher thermal maximum over time. But that just takes time for these things to establish, grow and things, but it probably will happen at some point. There's also a bunch of physics with the water. As soon as it gets hot, diffusion changes and things like this. And so there's all these other sorts of interesting effects that do happen when temperatures also increase. And so it can happen, does a whole bunch of stuff. And this is why we're also worried about corals and things like that. In Gulf of Mexico, is that they're basically beyond their thermal maximum that they can safely grow at.
Well, how can we keep cyanobacteria out of our lakes?
Oh, that's also one of those million-dollar questions, is that it's a fine balancing act, because we need these cyanos to do these very basic functions that they've been doing, like produce oxygen, cycle carbon and things like that for hundreds of millions of years. And it's just really these bad players that we want to exclude. And so there are some treatments that can happen. So if you have a big microcystis bloom that's persistent, things like hydrogen peroxide dumping, it takes a lot depending on the volume of the lake, but things like hydrogen peroxide treatments can actually help. Copper treatments can also help. And so this is like a whole other side of engineering research that's really like thinking more broadly about how do we manage these blooms once they actually start establishing. But those are two of the bigger ones. And it depends, again, on the species. So some species are more tolerant to, say, hydrogen peroxide than others. And then you also have to worry about your native species that you're killing off as well. And so it's definitely a big balancing act. But the first one would probably also be like reduce nutrients if you can.
Very good. I think we have time for one more question here. I'm hoping I'm going to pronounce this right. I think it's spelled out phonetically here. What's a eukaryote?
Oh, a eukaryote is us, right? So plants, fungi, protists, all these sorts of things that we see all this time with our naked eye a lot are all eukaryotes. And basically, a eukaryote is different than a bacteria because its cell type has several cell features like a nucleus, right? So bacteria don't have a nucleus. They don't have things like a Golgi apparatus. There's all these other different vacuoles and things that eukaryotes have. But the big defining factor, that's sort of the defining factor. But more largely, they get bigger, right? A lot of them are multicellular, while a lot of bacteria, while they tend to work together, are still considered single cells. So, good question.
Wow. Thank you, everyone, for all those great questions. And thank you, Dr. Sheik. All right, let's turn this back over to John. John?
Thanks, Bill, and big thank you to you, Cody, for all that great information. Cody is one of the few limnologists or lake ecologists I know who can look right down into the genes and figure out what's going on. Thanks for that. If you'd like to watch the program again or recommend it to your friends and neighbors, a recording of today's talk will soon be on our website at itascawaters.org. We'd love to have your feedback, so click on the survey link, which will be on the screen at the end of today's program, or you'll be getting an email link to the evaluation from Chad Manicky of Minnesota Sea Grant, so you can choose how you'd like to respond. We would really appreciate hearing from you. Our next program will be on September 7th, and our speaker will be Ann Nelson, public health educator with the Minnesota Department of Health. She will be speaking about the health of wells in Minnesota. I'm John Downing, and thanks so much for being with us today.